Okay, let's take a look at your lipids. And so your lipids, they are a diverse group of biomolecules. You have here, aside from what we learned already, you have your carbohydrates, your proteins, and so you have here your lipids. And so again, they are diverse biomolecules. And with that diversity, accordingly, this lipid, the term lipid, accordingly has an operational rather than a structural definition. Okay, and so your lipids, they are substances, and so that are found in living organisms, and so of course they are found in your membranes, cell membranes of your cell, in your cell membranes, and so with that they are lipids are found in organisms that are actually dissolved in nonpolar solvents. Okay, and so your lipids, they dissolve in nonpolar solvents. And so what are these nonpolar solvents? Examples of which of these nonpolar solvents that can dissolve your lipids, you have your ether. And so as in the laboratory, and so we used of your petroleum ether. Okay, and so you have your chloroform and your acetone. Okay, and so this ones, your ether, your chloroform, and acetone, they are nonpolar solvents, and they can dissolve your lipids. And so with that, and so your water is will not and so your water cannot dissolve lipids. Okay, and so your lipids, they are immiscible or insoluble with water. Okay, and so your functions of your lipids being actually diverse, and so includes what you call this your what you call this they include your structural functions, as which several types of lipids accordingly are important structural components in your cell membrane. And so with that, of course, they are found in your lipid bilayer, no? And then your fats and oils, and so they are what you call this uh energy storage. And then your lipid, and so your lipid molecules, and so that are found in the outer surfaces of various organisms, have protective waterproofing functions. Okay, and so with that, of course, for human beings, you have your glands. You have actually glands present in your skin that are capable of secreting oils, no? And so that is the what you call this that will what you call that will serve as a protective and waterproofing substances. Okay. Membranes. Okay, and so let's first take a look at your membranes. Okay, and so accordingly, most of the properties attributed to your living organisms. And so, what are these properties? Examples of which you have your movement, your metabolism, and growth and reproduction. And so, accordingly, these properties attributed to your organisms depend either directly or indirectly on your membranes. Okay, and so with that, accordingly, all your biological membranes, they have the same general structure. Okay, and so your membrane, they contain lipids and protein molecules. Okay, and so they contain lipids and protein molecules. Okay, and so with that, let's first take a look at your membrane lipids. Okay, and so when a hydrophobic, and so when a lipid molecule is actually suspended in water, and so when they are submerged in water, and so they rearrange to form ordered structures. Okay, and so they rearrange to form ordered structures. And with that, let's take a look at that ordered structure. Okay, and so your hydrophobic groups, those water-fearing groups, they are arranged in the interior, and so in the inside, no? And then your hydrophilic groups, and so they are exposed in your water, and so they are exposed in your water. Okay, and so they form actually a circle. And so with that, your hydrophobic groups will be at the interior, while your hydrophilic, and so there's a water loving, and so examples of which you have your phosphate, is they are exposed. Okay, and so this one, there's a membrane lipids accordingly, and so Okay, and so with that, your phospholipids accordingly form into biomolecular layers at a relatively low concentration. Okay, and so you have there again, and so your membrane lipids, and so when you submerge them in water or you suspend them in water, and so they rearrange to form ordered structures. Okay, and so your hydrophobic groups, and so will be at the exterior, while your hydrophilic group is exposed to your water. Okay, and so your membrane fluidity, and so this one is actually the fluidity or the movement actually of the membrane. And so with that, this one accordingly is determined by the percentage of unsaturated fatty acids in your phospholipids. And so note that your unsaturated fatty acids, and so they are not densely packed. No, and so they are not densely packed, and so that is why, and so you have here making the membrane uh, somewhat fluid, no? And so it is somewhat fluid, okay? And so that is for your membrane fluidity and your membrane lipids. Okay, moving on, let's take a look at your membrane lipids for your selective permeability. Okay, and so your membrane lipids, they function for selective permeability, and so that is the presence of your lipid that is due to the presence of your lipid bilayer in the membrane. And then, of course, your membrane lipids also functions for repair. Okay, and so let's take a look at your selective permeability. Okay, and so that is you have your hydrophobic hydrocarbons present in your lipid bilayer provide a barrier for the transport of polar. This barrier is somewhat impermeable or impenetrable barrier. And so for the transport of your polar and your ionic substances. And so with that, and so, but then of course, and so selective permeable, and so you have it will what they call this, it will only select those, uh, it will allow the pass of it will allow your what you call this a non-ionic and non-polar substances to pass through the membrane, of course, with that. And so for your polar and your ionic substances, and so you have other mechanisms for them to what you call this to enter the membrane, and so to cross the membrane. And then you have your repair. Okay, and so accordingly, when a lipid bilayer are disrupted, and so you have here the capacity of your membrane to repair. And so they can, and so when there is a cut, and so or a damage, and so there's the your membrane is capable of doing a reseal or repair, and so of your damaged part. Okay, and so accordingly, and so your small plasma, and so plasma, if you have a small plasma membrane, and so breaks spontaneously, and so that is actually sealed via your lateral flow of lipid molecules. Okay, and so while for your what you call this a repair of your uh, larger lesions, and so caused by your mechanical stress and mechanical trauma, and so that is it requires what you call this an energy and calcium. And so that is again your membrane, and so your membrane lipids, and so or your membrane, and so they are capable of and so the function, sorry, for selective permeability and for repair. Okay, and so your brakes for your repair, and so your brakes are sealed via lateral flow of lipid molecules. Okay, and so let's take a look at your proteins, membrane protein. Okay, and so your membrane proteins. And so most of the functions associated with biological membranes require protein molecules. Okay, and so your membrane proteins, accordingly, they are classified by the function they perform. Okay, and so you have your structural membrane proteins, you have your membrane proteins that functions for transport. You have your uh, those that function for catalysis, for signal transduction, or for immunological function. Okay, and so they are also accordingly classified to their structural relationship with the membrane. You have there with that, you have your two, you have your peripheral and your integral proteins. Okay, and so you have your peripheral and your integral proteins. Okay, and so your peripheral, of course, and so that is at the periphery, while for your integral, and so that is integrated in the uh, membrane. And so you have here an illustration of your peripheral and your integral proteins. Okay, and so that is for your membrane proteins. Of course, for your membrane. And so let's move on to your lipids. Okay, and so your lipids, and so your lipids can be classified as, and so you have your fatty acids, triacylglycerols, you have your wax esters, your phospholipids, your sphingolipids, and your isoprenoid. Okay, and so these are actually accordingly your six actually six classes of your lipids. Okay, and so aside from you have your simple, derived, and your compound lipids, you have this as classes also of your uh, lipids. Okay, and so your fatty acids, and so they are monocarboxylic acids that what they call this typically contain hydrocarbon chains with 12 to 20 or more carbons. Okay, and so you have here with a 12 to 20 or more carbon atoms in the chain. Okay, and so this in number your carbon atoms accordingly are numbered from the carboxylate group, and so this one is for the nomenclature. No, and so the number your carbon atoms are numbered from your carboxylate end, and so you will start numbering from your carboxylate end. And so let's take a look at for in this example, and so this one is your carboxylate end, and so you start numbering, and so your uh carbon number one is your uh, carboxylate carbon. And then you follow, and so you follow with that. Okay, and so accordingly, you have here for your Greek letters accordingly are used to designate a certain carbon atoms. And so with that, you have your alpha carbon is adjacent to the carboxylate group. And so it is just situated just next to your carboxylate group. Okay, and so it is denoted by, and so you have here as your first carbon. And so if you follow your IUPAC naming, and so as 1, 2, 3, 4, and the following, and so this one is at the second carbon. And so your alpha carbon is your number two, carbon number two. Okay, and so and then your omega, and so and then as it will follow, and so your beta, and then it will follow. With that, your omega carbon, and so that is your terminal methyl carbon. Okay, and so let's take a look at this one, and so you have it as a CH3, and so that is a methyl one. No, ayan. So you have that as for your omega carbon. Of course, that is denoted by that sign, no? Ayan. So this, what you call this fatty acids. And so your fatty acids, they are important components of your several types of lipid molecules. And they are primarily found in your triacylglycerols and membrane-bound lipid molecules. Okay, and so that is for your fatty acids. And so let's take a look at that fatty acid. Most naturally occurring fatty acids have an even number of carbon atoms, even number. And so you have your 18, and so your 2, 4, those accordingly, they are your most naturally occurring fatty acids. Okay, and so you have there. And so, and that, of course, they form an unbranched chain. And so they form an unbranched chain. And so your fatty acids that contain only carbon to carbon single bonds, they are known as your saturated fatty acids. And so you have your single bonds. In your saturated fatty acids, there is no double bond present. You only have single carbon to carbon bonds, no? And so while in your unsaturated fatty acids, you have one or more double bonds present in the chain. Okay, and this double bonds, and so with this double bonds, they can be in your cis and trans forms, no? And so they are your isomeric forms of these double bonds. Okay, and so again, your double bonds accordingly are rigid structures so that molecules that can contain them occur into isomeric forms, no? And so that is your cis and trans. Okay, And so you have here a table of your some common fatty acids, no? And so you have your saturated here, you have your myristic acid, and so your palmitic, your stearic acid, your arachidic acid, your lignoceric acid, and your cerotic acid. And so please don't confuse your arachidic acid with arachidonic acid, because your arachidonic acid here is unsaturated, no? And so your arachidonic is unsaturated. And so with your unsaturated fatty acids with that, and so you have your palmitoleic acid, oleic acid. This one actually is the most common, no? And so your linoleic acid, linolenic, and then your gamma linolenic, and then your arachidonic acid. Okay, and so these are examples of your common fatty acids, saturated and unsaturated fatty acids. And so with the presence of your double bonds, and so for your unsaturated fatty acids, let's take a look. And so you have your oleic acid, and so you have under your oleic acid, and so you have your myristoleic, and so it has this oleic acid series, and so they contain one double bond. And so there's a myristoleic, palmitoleic, oleic, gadoleic, and your nervonic acids, and so they contain one double bond only. And so one double bond, one carbon to carbon double bond in the chain. And so again, ulitin ko, they contain your oleic acid series, they contain one carbon to carbon double bond only. Okay, and so with that, and so these are your corresponding number of carbon atoms present, and they are found in these oils. Okay, and so your myrosilaic, that is found in your butter, your fish liver oil, and your sperm oil. Okay, and so your palmitoleic, and so in your butter, fish oil, and your hen fat, no? And so your oleic, and so they are found in plant and animal fats, and then your olive oil. Okay, and so let's take a look at your linoleic. And so your linoleic, it contains two carbon to carbon double bonds. No, and so your linoleic, it contains two carbon to carbon double bonds. It has 18 carbon atoms, and so that is found in your linseed oil, lard, cottonseed, and cottonseed oil. No, and so they are largely found in your linseed oil, lard, cottonseed oil. And then you have your three carbon to carbon bonds, double bonds, and so you have your linolenic acid, okay, and so at 18 carbon atoms still. And so this is largely found in your linseed oil, and then your fish liver oil, and then your arachidonic acid, and so that contains four carbon to carbon double bonds, no, and so it has 20 carbon atoms, and that is found in your lecithin, cephalin, butter, and liver fats, and then your clupidonic acid, and so that is it has five carbon to carbon double bonds, and so that is found in your brain and liver phospholipids. Okay, and so that is for your for your unsaturated fatty acid. So still under your unsaturated fatty acids, you have your cis and trans isomer. Okay, and so your unsaturated fatty acids again, and so they have your cis, they have these isomers, you have your cis and trans, okay? And so with your cis, and so they are, the identical groups are on the same side of the double bond. And so let's take a look with this. And so they are the same, on the same side of the double bond. While for your trans isomer, and so the groups are on the opposite sides, no? And so take a look at the illustration here. And so this one, and so on your right side, and so that is for your trans isomer, and the one on the left side, and so that is for your cis isomer. Okay, and so for your cis isomer, and so your cis configuration, and so do not pack closely together. Okay, and so they do not pack closely together. While, and so your what do you call this, your trans isomer, and so your lesser energy is required to break your intermolecular forces between your unsaturated fatty acids. And so remember that your unsaturated fatty acids accordingly, and so they occur in your cis isomer. And so according most occur in cis isomer. And so with that, your unsaturated fatty acids do not pack as closely together as your saturated fatty acids. Owing, of course, and so when they do not pack, and so there is a somewhat, there is an easy movement, no? And so that is for your unsaturated fatty acids, okay? And so they do not pack closely together, and so giving you, and so in your membrane fluidity of your membranes, and so that is accordingly somewhat because of your unsaturated fatty acids present again, and so they do not form a rigid structure, and so you have there for your cis and your trans ones, okay? Ayan, so you have for your trans isomers, ayan, so accordingly, ayan, so they have a lower melting point and they are liquids at room temperature. And so the double bonds, again, ayan, so in your naturally occurring fatty acids, they are in your cis configuration. Okay, and so they are in your cis configuration. Okay, and so that is for your cis and trans isomer. Okay, and so your trans double bond and so possess a three-dimensional structure. Okay, and so your trans double bonds and so have three-dimensional structures similar to those of saturated fatty acids, no? And so you have here, and so your presence of one or more double bonds, and so that it will make accordingly your uh, the unsaturated fatty acids susceptible for oxidation. Okay, and so they are susceptible for oxidation, and of course, and so the consequence, the effects, and so of this oxidative stress, and so on your cell membranes, your consequences of this sorry consequences of this oxidation or include the effects of your oxidative stress, of course, on your cell membranes, and the tendency of your oils to become rancid. Okay, and so you have here. Okay, and so accordingly you have your monounsaturated fatty acids here, and so they contain one double bond present, one carbon to carbon double bond present in the chain. Okay, and so when you have and so examples of this, you have your oleic acid as what we've seen in the table earlier, your polyunsaturated fatty acids, and so they contain two or more double bonds, and so two or more carbon to carbon double bonds separated by a methylene group. Okay, and so examples, you have your linoleic acid, and others. Okay, and so you have there, and okay, and so accordingly, and so there's a linoleic, and so your linoleic and your oleic, and so they are what you call this, the most abundant fatty acids in your living organisms. Okay, and so let's take a look at your essential and non-essential fatty acids. Okay, and so your organisms like your plants and bacteria together, your organisms like your plants and bacteria, and so they can synthesize all the fatty acids they require from your acetyl-CoA. But then for your mammals like your humans, and so they obtain most of their fatty acids from your dietary sources. Okay, and so with it, these fatty acids that are obtained from the diet, you have it as your essential fatty acids. Okay, and so these essential fatty acids, and so these are fatty acids that cannot be synthesized by humans. And so the required enzymes for the synthesis of these essential fatty acids is actually absent in humans. No? And then you have your non-essential fatty acids. Your non-essential fatty acids, and so they say fatty acids that can be synthesized by mammals. So, yan. So, of course, by your humans, no? And so you have there. Okay, and so you have there for that. Okay, and so your essential fatty acids. And so your essential fatty acids, they are used as structural components. And so like your phospholipids that are present in your membrane. Again, your essential fatty acids are used as structural components and as precursors for several important metabolites. Okay, and so these metabolites include your eicosanoids and your anandamine. Okay, and so anandamine. Okay, and so that is for that. Of course, your eicosanoids, and so these are produced in your human tissues, and so that include your prostaglandins, your thromboxanes, and your leukotrienes. And so these eicosanoids, and so mediate wide variety of physiological processes, which include your inflammation, mediation of inflammation, smooth muscle contraction together with your blood flow regulation and perception of pain. Okay, okay, and so that is for that. Okay, and so you have your omega-6 fatty acids. Your omega-6 fatty acids here, and so these are your linoleic acid in its and its derivatives, no? And so your omega-6 fatty acids. Okay, and so your linoleic acid in its derivative. Of course, with this, and so sources of your omega-6 fatty acids, and so you have your vegetable oils like your sunflower and soybean oils together with your eggs and your poultry accordingly. And so that is for your essential and non-essential fatty acids. Have your omega-3 fatty acids. Your omega-3 fatty acids, and so you have here your alpha linolenic acid and its derivative. Okay, and so your omega-3 fatty acids, you have it as your alpha linolenic acid and its derivatives. And so with this, like example, your derivatives of your alpha linolenic uh, fatty acids, you have here your docosahexaenoic acid, and so your docosahexaenoic, sorry, and so with an E, docosahexaenoic acid, and your eicosapentaenoic acid. Okay, and so you have your eicosapentaenoic acid and your docosahexaenoic acids, and so these are actually referred to as your omega-3 fatty acids. Okay, and so referred to as your omega-3 fatty acids. Okay, your eicosapentaenoic acid here and your in your docosahexaenoic acid, and so they are what they call they are believed accordingly to promote cardiovascular health, and they lower blood triacylglycerol levels, and then you have your the lower blood pressure, and they also decrease platelet aggregation. Okay, and so that is for your eicosapentaenoic acid or your docosahexaenoic acid as or commonly referred to as your omega-3 fatty acids. Okay, and so sources of your omega-3 fatty acids or your sources of your alpha linolenic include your flaxseed, and so your soybean oils, your walnuts accordingly. And so you have your sources, you have your flaxseed, your soybean oils, and walnuts. Okay, on to the next class of lipids, and so let's take a look first at your eicosanoids. And so your eicosanoids, and so they mediate physiological processes like, and so as what we said earlier, muscle contraction, inflammation, please put a comma here. Okay, pain reception and blood flow regulation. Okay, and so with this, your eicosanoids accordingly are implicated in several diseases like your myocardial infarction and rheumatoid arthritis. Okay, and so these eicosanoids, they are generally active within the cell, the cell that in which they are actually produced. Okay, and so they say with that, and so because they are generally active in the cell that are in which they are produced, and so these eicosanoids are uh, otherwise known as autocrine regulators. But then, and so for that, for uniformity, and so let's use your eicosanoids. Okay, and so your eicosanoids. Okay, and so these eicosanoids, and so they are derived either from your arachidonic acid or your eicosapentaenoic acid. Okay, and so they are derived from your arachidonic acid and your eicosapentaenoic acid. And so later, and so you will see you will see your AA, and so you have here AA. This one means arachidonic acid, no? And then your EPA, you have your eicosapentaenoic acid. And so please don't be confused with those. Okay, and so your eicosapentaenoic acid, and so they are derived from either, and so but either your arachidonic acid or your uh eicosapentaenoic acid. Okay, and so your production of your eicosanoids accordingly begins when your arachidonic acid or your eicosapentaenoic acid is released from phosphoglyceride by your phospholipase A2, no, or your phospholipid. Okay, your production begins when your phosphoglyceride is released or your phospholipid is released by your phospholipase A2. Okay, and so you have your eicosanoids, you have your prostaglandins, your leukotrienes, and your thromboxanes. Okay, let's take a look at your prostaglandins. Your prostaglandins, and so they contain a cyclopentane ring and a hydroxyl group. And so the hydroxyl group accordingly is located at C1, C11, sorry, carbon 11 and carbon 15. And so, but then for that, the important thing is there is the presence of your hydroxyl group. Okay, and so the most important of your prostaglandins accordingly are those that are derived from your arachidonic acid. And so you have actually two, and so two series that are derived from your arachidonic acid. And so you have two series that are derived from your arachidonic acid. While for your EPA, for your eicosapentaenoic acid, you have three series of prostaglandins produced from that. Okay, and so your prostaglandins, and so they are involved in inflammation, and so your prostaglandins, they are involved in inflammation. And so your inflammation, this one is a mechanism to fight an infection process, no? And so this, your inflammation, this one is what you call this, a mechanism to fight an infection. Okay, and so with this, in your inflammation, there is a, in the process of fighting an infection, and so it produces pain and fever. Okay, and so that is for your inflammation. Okay, and so there is the production of pain and fever. And so with this, again, your inflammation, this is a mechanism of the body to fight an infection. And then your prostaglandins, and so they are involved also in reproduction. Okay, and so in specifically in your ovulation and uterine contractions during conception and labor. Okay, and so and you have here for your, they also these prostaglandins are also involved in digestion. And so with that, and so an example of your involvement of your prostaglandins in digestion is it inhibits your gastric secretion. Okay, and so inhibits gastric secretion. Okay, and so that is for that again. And so your what you call this, your prostaglandins, and so the biosynthesis of your prostaglandins begins once your arachidonic acid has been released from the membrane phosphoglyceride. Okay, and so of course you have two series again from your prostaglandins, and you have three from your EPA. Okay, and so that is for that. Okay, and so arachidonic acid is first converted to your what you call this, arachidonic acid is converted to your prostaglandin H2, and so it is from your, and so your arachidonic acid or your EPA is released from your phospholipid or your phosphoglyceride, and then your arachidonic acid is first converted to your prostaglandin H2 by your enzyme cyclooxygenase and then your peroxidase. Okay, and so you have here your arachidonic acid is converted to your prostaglandin H2 and and then catalyzed by your enzyme cyclooxygenase. And your prostaglandin H2 is converted to your prostaglandin E2 by your prostaglandin endoperoxidase isomerase. Okay, and so you have here your prostaglandin endoperoxidase isomerase. And this prostaglandin E2, and so this one induces fever and it will induce fever. Okay, and so that is for your prostaglandin E2. Okay, and so just for your information, you have actually a drug, your aspirin, and so accordingly, it is a what you call this, a pain reliever, and what you call this, anti-inflammatory. I mean, antipyretic, accordingly. And so it is your aspirin, and so it relieves minor pain and it reduces fever and inflammation. And so with that, what is the action of your aspirin? And so that is, it will inhibit or inactivate, hence your aspirin will inactivate your cyclooxygenase. And so it will inactivate your cyclooxygenase. How will it inactivate? By acetylating a serine residue in the enzyme's active site. Okay, but then with that, your aspirin, and so it is important for you to know that your aspirin, it relieves pain and it reduces fever and inflammation by, what you call this, how will it do that is to inactivate your cyclooxygenase. And then you have your leukotrienes, and so you have your leukotrienes, and so these are linear molecules, and so they are linear molecules. And so the synthesis of your leukotrienes is initiated by peroxidation. Okay, and so that is initiated by peroxidation. Ayan, so by peroxidation reaction. Okay, and so that is catalyzed by lipoxygenase. Okay, and so your leukotrienes, and so they are powerful chemotactic agent, and so they are powerful chemotactic agent, and so there's a chemotactic agent, and so they attract immune cells to the site of tissue injury, and so to the site of tissue injury, and so also, and so they say leukotrienes, and so they are involved, they induce vasoconstriction and bronchoconstriction, and so there is a what we call this narrowing of the blood vessels and narrowing of your uh, bronchi. And so with that, and also, and so with this, and so your leukotrienes accordingly, and so some leukotrienes, and so are identified as a slow reacting substance of anaphylaxis. Okay, and so they are slow reacting substance of anaphylaxis. And so your anaphylaxis here, and so this is actually a severe allergic reaction. Okay, and so of course your anaphylaxis, and so in this, and so your chemotaxis and the rest, and so you will learn more of this in your uh, serology, immunoserology. Okay, and so moving on, you have your thromboxane A2, and so your thromboxane, sorry, your thromboxane, and so this is the last of your eicosanoids, no? And so your thromboxanes, and so they are derivative of your arachidonic acid or your uh, eicosapentaenoic acid. Okay, and so this is a cyclic, and so this is a cyclic. It has a cyclic structure. Okay, and so your thromboxane A2, and so is produced from arachidonic acid by platelets. Okay, and so your thromboxane A synthase converts your Okay. And so your thromboxane A synthase, it will convert your prostaglandin H2 to your thromboxane A2. And so recall that you have your prostaglandin, and so from your arachidonic acid to your prostaglandin H2 in your prostaglandins. Okay. And so with that, your thromboxane A2 is produced from your prostaglandin H2 by your enzyme thromboxane A synthase. Okay, and so with that, and so this thromboxane A2, and so promotes platelet aggregation and vasoconstriction, no, following a tissue damage. Okay, and so you have here, and so it will promote platelet aggregation following a tissue damage. Okay. Ayan, so with that, activation of your platelets releases thromboxane A2. Ayan, so with that, and so it will to promote aggregation and vasoconstriction. And so after a tissue damage, again, so you have there, and so of course to prevent, and so massive blood loss, and so those items. Okay, and so your thromboxane A2 accordingly is rapidly converted to into its inactive form. Ayan, so inactive metabolite. Ayan, so TXB2 by an isomerase. Okay. And so the second class of your lipids, you have your triacylglycerol. And so you have your triacylglycerol or your triglyceride. Okay, and so they are your esters, your triacylglycerol, and so they are esters of glycerol with free fatty acid molecules. Okay, and so you have your, again, you have your glycerol ester with three fatty acid molecules, no? And so your monoacylglycerol, ayan, so this one is a glyceride with one fatty acid group. Actually, this one is your metabolic intermediate, no? Your monoacylglycerols and your diacylglycerols. Okay, and so with this, and so they are normally present in small amounts. Okay, and so you have here, and so your triacylglycerols, they do not possess a net charge, and so or they have no charge, no, and so they have no charge, and so that is why they are referred to as neutral fat. And so again, your triacylglycerols, and so they do not have a charge, and so that is why they are referred to as neutral fat. Okay, and so that is neutral fat. Okay, and so accordingly, most triacylglycerols or triglyceride molecules contain fatty acids of varying length. And so these fatty acids, and so are either saturated, unsaturated, or a combination of both of these. Okay, ayan, so you have there. Ayan, so your mixtures of your triacylglycerol are referred to as your fats and your oils. So, no, and so fats or oils. Okay, and so again, ayan, so your mixture of your triacylglycerol, ayan, so they are referred to as fats or oils, no? Ayan, so fats or oils, and so it depends accordingly on their fatty acid composition. And so you call it as a fat, and so if you have a larger proportion of saturated fatty acids, and these fats, they are solid at room temperature. Ayan, so while, ayan, so sorry. And so let's take a look at this one. And so uh an illustration of your triacyl glycerols. And so note accordingly that you have your and so triacyl glycerol. And so you have the glycerol one, and then you have the fatty acid. Okay, and so you have your triglyceride, and so you have when you hydrolyze this one, and so you have your glycerol and fatty acid. Okay. And so that is upon hydrolysis, of course, it will produce you such, no? Let's go back to your fats and oils. Your fats, and so your fats, they are solid at room temperature, and they contain a large proportion of saturated fatty acids, no? While for your oils, and so they have a high unsaturated fatty acid content, and they are liquid at room temperature. Okay, and so they are. they have a high unsaturated fatty acid content and they are liquid at room temperature. Okay, and so your triacylglycerols, and so they have several functions. Okay, and so with that, they are actually your first on the list accordingly, and so it is actually the major storage and transport form of fatty acids. Okay, and so you have here your this one, your triacylglycerols, and so they are uh, triacylglycerol accordingly, and so they are more efficient in storing energy than your glycogen. And so they are more efficient in storing energy than your glycogen, no? Okay, and so accordingly, your triacylglycerols are hydrophobic, no? And so you have here, and so therefore, accordingly, and so they coalesce into compact anhydrous droplets within cells. And so they are not insoluble. That is why they will become anhydrous, no? And so you have here your triacyl anhydrous triacylglycerol, of course, it's stored by your adipocytes in the adipose tissues. So, no, and so your the stores are actually equivalent amount of it stores equivalent amount of energy in about 1/8 of the volume of a glycogen. And so you have there. And the other reason, as a more giving you a more efficient storing energy storing capacity or energy storing property is you have your triacylglycerols are more reduced. And with that, and so release more electron atoms when oxidized. And so than an equivalent amount of carbohydrates. Again, and so your triacylglycerols are more reduced and can uh, release more electrons when oxidized. Okay, and so accordingly, and so your triacylglycerols and so release more energy and so when they are degraded, and so comparing it to your carbohydrate. Okay, and so that is it for your uh, energy storing capacity, ability of your triacylglycerol. Third, and so you have your function still. You have there as a what they call this you have the function for storage, and then with this and so your triacylglycerols, and so uh they provide insulation and so in low temperatures. And so note that your fat and so your triacylglycerols and so or your fat and so they are uh poor conductors of heat, and so that is why and so they prevent heat loss, and so your fat prevents heat loss. Okay, and so your adipose tissue, and so because with its high triacylglycerol content, and so they are found in your uh throughout the body, no, and so especially underneath the skin. Okay, and so uh you have here for your it provides insulation and insulation at low temperatures. Okay, and then other than that, and so that is an energy reserve in plants. And so accordingly, your triacylglycerol, and so that a what they call this constitutes an energy reserve in fruits and seeds accordingly. And so from in your peanuts, in your corn, in your palm, in your soybean, in your olives accordingly. And so this accordingly, and so they contain large amount of unsaturated fatty acids. Okay, and so you have there for your plant oils, no? And so okay, and so you have again, and so your in plants, and so you have your triacylglycerol, and so they what they call this, they are energy reserve in plants. Okay, and so accordingly, because these molecules contain relatively large amounts of unsaturated fatty acids like your oleic and linoleic acids, and they are referred to as your plant oils, no? And so you have your seeds that are rich in uh, oil, and so include your peanut, your corn, your palm, your soybean, your olives, your avocados, and your olives. Okay. And then you have your glycerol. And so let's take a look first at your glycerol. And so before we move on to the next class. And so when degraded, and so your triacyl glycerol, and so when degraded, and so it will produce your fatty acids and your glycerol. Okay. And so your glycerol, actually, this one is your simplest trihydric alcohol, no? And so this one is commonly known as your glycerin. And so this one is commonly known as your glycerin. And so you have here, it is oily and so colorless, heavy, liquid with sweet taste. And so I don't know, I haven't tasted this in the laboratory, no? And so but then, and so for the literature, and so it has a sweet taste, no? And so it is the component of fat. Please change this one. And so the component of fat responsible for a positive acrolein test. And so when you do your acrolein test, and so the positive result is an acrid or pungent odor accordingly. Okay. And so in the body, and so your free glycerol is absorbed and metabolized. And so and this one of course, and so this free glycerol, and so it produces caloric value same with your glucose. So okay. Ayan, so you have there. And oxidation with your hydrogen peroxide in alkaline solution in the presence of your iron. Iron salt will produce aldehyde and dihydroxyacetone. Okay, and so treatment with nitric acid will form glycerin, and so treatment with your nitric acid will form nitroglycerin, and so this nitroglycerin is used in the manufacture of dynamites and smokeless powders. Okay, and this one, other than that, and so it is used, it is your glycerol. This one is a byproduct in the manufacture of your soap, and this one is used in the production of cosmetics. And so, accordingly, due to its solubility and hygroscopic nature, and of course, your nitroglycerin, other than it being used as in the manufacture of your dynamites and smokeless powders, and so it is used as vasodilator in treatment of hypertension. Okay. Okay, and so in your triacylglycerol, and so you have here your fats, and so you have your animal fats, and so it contains mostly of your oleic, palmitic, and your stearic acid, your mutant fat, more of your stearic and your less oleic than pork, and then you have your butter fat, and so composed mainly of palmitic, oleic, and your some amount of butyric and your caproic, and then your human fat, and so they are mostly oleic and they have a yellowish tinge due to carotene and xanthophylls pigment derived from your ingested foods. Okay, and so examples of your uh, triacyl glycerols accordingly. Next class, you have your wax esters. And so your waxes, and so your wax esters or your waxes, they are complex mixtures of nonpolar lipids, no? And so they are what they call this, they are composed of long chain fatty acids and long chain alcohols, no? And so uh your wax esters. Okay, and so your wax esters, they are composed of long chain fatty acids and long chain alcohols. And these wax esters, and so they are component of your waxes. Okay, and so you have there and your waxes, and so they are protective coatings on your leaves, your stems, and fruit of plants, and on the skin and fur of animals. No, and so your wax esters, your wax, okay, and so your wax, well-known waxes, you have here your carnauba wax from leaves of your Brazilian wax palm. And so this, and so it contains melicyl cerotate, no? And then you have your beeswax, and so this one is, it contains ayan, so it contains triaconyl hexadecanoate. And so this one, ayan, so they are your, what they call this, your waxes also contain hydrocarbons, again, as what we said, hydrocarbon and long chain fatty acids, and aldehydes accordingly. And so together with your sterols. And so, but then with that, and so that is for your wax and wax ester. Next class, you have your phospholipids. Your phospholipids, and so they are your structural components of membranes. And so, of course, other than that, and so it possess other roles in living organism. No, and so. Of course, the first one is your structural components of your membranes. And so, other than that, and so uh, it functions as an emulsifying agent and surfactants. And so, your surfactant here, or your surface active agent, is a substance that lowers the surface tension of a liquid, and so that it will spread out over a surface. Okay, and so you have there your phospholipids, and so they are amphipathic molecules, no, and so they are amphipathic molecules. They have your hydrophobic and your hydrophilic domains present. Okay, and so what you call this, your phospholipids, and so your phospholipids, and so they possess your hydrophobic domain and your hydrophilic domain. And so your hydrophobic domain, and so that is your hydrocarbon chains of your fatty acids. No, and then your hydrophilic one, and so that is your phosphate and your other charge polar groups. Okay, and so accordingly, when you suspend it in water, and so that is same as what we said earlier, and so when you suspend this phospholipid in water, or you submerge, and so they rearrange into ordered structure. Okay, and so they rearrange into ordered structure again. And so as what you already know, and so the hydrophobic domain, and so will be buried in the interior, in the yes, in the interior part, and then your hydrophilic domain, and so the phosphate, and so is at the exterior part, having contact with the water. Okay, and so again, and so this structure, and so is for your uh actually is the basis for your structure of your membrane. Okay, and so this is your hydrophilic, and this one is your hydrophobic. Okay, and so when you submerge, and so this is actually the ordered structure, and so that will happen when you submerge a phospholipid in a water. Okay, and so and so we take a look, and so they are buried in the interior, and those what they call these polar groups, and so are at the exterior. Okay, and so this one again, when you suspend a water, I mean a what they call this, a phospholipid. When you suspend a phospholipid in a water, and so they rearrange to produce an ordered structure. Okay, and so the hydrophobic groups of your phospholipids are buried in the interior, no? And so to exclude water. And so while your hydrophilic, and so your polar head groups are exposed in the water, no? Okay, your phospholipids, and so your there are actually two types of your phospholipids. You have your phosphoglyceride and your sphingomyelins. Okay, and so your phosphoglycerides, and so they contain glycerol, fatty acid, phosphate, and alcohol. While your sphingomyelins, and so it has a sphingosine. It contain also your glycerol, fatty acid, and phosphate, but instead of alcohol, and so you have a glycerol. Okay, and so your this one, your sphingomyelin, and so they are what they call they are insoluble, no, in cold alcohol, in so but they are soluble in chloroform, hot alcohol, and benzene. Okay, and so just a clinical significance of your sphingomyelin, and so in case of your Niemann-Pick disease, and so there is a deposition of sphingomyelin in the liver and in the spleen. Okay, your sphingomyelin, and so let's take a look first at your sphingomyelin. And so your sphingomyelin, and so this one is found in most animal cell membranes. Okay, and so found in most animal cell membranes, and this one is in the what they call this, they are found in your myelin sheath of nerve cells. Okay, and so in the myelin sheath of nerve cells, and so the myelin sheath, and so is formed by successive wrapping, and so successive wrappings of a cell membrane of your specialized myelinating cell called as your axon. And so this one, and so the same myelin sheath facilitates for the rapid transmission of impulses. And so again, your insulating properties of your sphingomyelin, and so facilitates the rapid transmission of your nerve impulses. And so this one is an illustration of a myelin sheath. No, and so you have your sheath here. Okay, and so you have a successive wrapping. Your phosphoglyceride, and so your phosphoglyceride, and so they are the most numerous phospholipid molecules that are found in the cell membranes. Okay, and so your simplest phosphoglyceride, your phosphatidic acid, actually is a precursor of all other phosphoglyceride molecules. No, and so your phosphoglyceride, and so they are classified according to the alcohol that becomes esterified in your phosphate group. Okay, and so example of which you have your phosphatidylcholine, and the esterified alcohol is your choline accordingly. Okay. And so with that. Okay, and so your fatty acid present in your phosphoglyceride, and so the most common, and so you have your 16 to 20 carbon atoms, no? And so at your carbon 1 of your glycerol, and so that you have your the attachment of your fatty acid, and that you're at your carbon 2, your substituent is your unsaturated fatty acid. Okay, and so that is for your phosphoglyceride. And so let's take a look at your other phosphoglyceride. Have here your phosphoglyceride and your what they call this hydroxyl group. Now you have your phosphatidic acid, and so that is with water. And so this one is the simplest of your phosphoglyceride. Your phosphatidylcholine, and so or your lecithin accordingly, and so that is with your choline as the hydroxyl group present. And then your ethanolamine, also known as your cephaline. And so you have your ethanolamine there, your phosphatidylserine, you have your hydroxyl group present, XR, your hydroxyl group present, you have your serine, and then your phosphatidylglycerol, you have the glycerol one, and then you have your diphosphatidyl or your cardiolipine, you have the cardiolipine accordingly. And then you have your phosphatidyl inositol, and so as the inositol. Okay, and so that is for your examples of your phosphoglyceride and the polar groups present. Your lecithin, this is found in your egg, liver, and nervous tissues. Okay, and so your fatty acid chains, and so you have your saturated, unsaturated fatty acids, or both. No, and so your freshly prepared lecithin accordingly is wax-like, and it turns brown upon oxidation. And so, of course, upon exposure to air, and so it will turn brown, no, and so and then they are this lecithin, they are soluble in organic solvents, and they produces acrolein, and so when heated with a dehydrating agent. Okay, and so your hydrolysis of your lecithin will give two fatty acid molecules and one phosphoric acid, one glycerol, and one choline. And so that is for your lecithin. your lecithin, and so your lecithin, when hydrolyzed by your lecithinase, will produce lysolecithin. Okay, and so that is your lysolecithin. Your lysolecithin here, and so this one is has a hemolyzing effect on RBC. Okay, and so please note that your lecithinase, this one is found in the venom of your cobra and spiders. And so that is why, accordingly, when you are beaten with one of these, with your cobra or your spiders, and so that possesses your lecithinase, and so you will have your RBCs are hemolyzed. Okay, and so your cephalin, and so this one is a your ethanolamine, no? And so this one is insoluble in ether and methanol, and it initiates the process of coagulation. And so with that, and so it is a thromboplastic substance. And your cephalin, as it initiates the process of coagulation, that is referred to as a thromboplastic substance. And then your phosphatidylserine, and so it contains serine, and this one is found in the brain tissue, and they are found in small amounts in the blood. Sphingolipids, and so your sphingolipids, and so they are actually important components of animal and plant membranes still, and so all, and so they contain a long chain amino alcohols, no? And so in animals, accordingly, and so in animals, this alcohol is primarily your sphingosine. Okay, and so alcohol present is primarily your sphingosine. Okay, and so you have there. And so the core, accordingly, of each type of your sphingolysine is your ceramide. No, and so the core is your ceramide. Okay, and so that is your amide derivative of sphingosine. Okay, and so you have here your glycolipids, and so you have your cerebrosides, your sulfatides, and your gangliosides. Okay, again, and so your glycolipids, or you have your glycolipids are actually lipid molecules with carbohydrate groups attached, you know. And so with this, your glycolipids include your glycosphingolipids, and so which will fall under your sphingolipids. And these glycosphingolipids include your cerebroside, your sulfatides, and your ganglioside accordingly. And so this one, and so there's a glycosphingolipids, they contain ceramide, and so but they differ from sphingomyelin, no? And so they differ from sphingomyelin in the content, I mean, because they do not contain phosphate again. And so your glycosphingolipids contain ceramide, but then, and so they differ from sphingomyelin, and so because they do not contain a phosphate. Okay, and so let's take a look at the gangliosides, your cerebrosides, and your sulfatides. Okay, and so your cerebrosides, they are sphingolipids, in which and so the head group is monosaccharide. Note that these are glycolipids, and so you have there. And so with this, and so your monosaccharide head group, and so is present in your cerebrosides, and so accordingly you have your galactocerebroside, that is the most common example of your cerebroside. And so this galactocerebroside is found entirely in cell membranes of the brain. Okay, and so found entirely in the cell membranes of the brain. And if your cerebroside accordingly is sulfated, and so it is referred to as your sulfatide, no? And so it is referred to as your sulfatide. With that, and so your sulfatides are negatively charged at a physiological pH. Okay. And so accordingly, and so your sphingolipids possess oligosaccharide groups. Ayan. And so, and these oligosaccharide groups accordingly contain one or more sialic acid residues. Ayan, so you have there. Okay, and so with this, and so your sphingolipids that contain oligosaccharide with one or more sialic acid residues, and so they are referred to as your ganglioside. Again, and so your ganglioside, and so these are sphingolipids that contain oligosaccharide groups with one or more sialic acid residues. Okay, and so and then of course your glycolipids, and so your glycolipids there, and so they bind your what you call this bacteria and bacterial toxins to your cell membrane, no, and so uh your glycolipids still, and so your bind bacterial toxins and your back as well as your bacterial cells to the cell membrane. Okay, and so you have therefore your cerebrosides, you have your galactocerebrosides, you have your sulfatides, and your gangliosides, no. And so with this, of course, you have therefore your uh, for your clinical significance of this, and so you have your Gaucher's this disease with this, no? Then you have your isoprenoids. And so you have your isoprenoids, and so they are biomolecules that contain repeating 5 carbon structural units known as your isoprene units. Okay, and so your isoprenoids, they contain repeating 5 carbon units known as your isoprene units. Okay, and so they are actually not synthesized from isoprene. And so your isoprenoids are not synthesized from isoprene or your methylbutadiene. No? And so they are what you call this? They are formed from isopentenyl, and so isopentenyl pyrophosphate from acetyl coenzyme A. Okay, and so they say isoprenoids consist of your terpenes and your steroids, no? And so they are uh they consist of your terpenes and steroids. And so your terpenes are the numerous groups of molecules that are found largely in your essential essential oils of plants, while for your steroids, and so are derivatives of hydrocarbon ring system of cholesterol, no? And so your terpenes and so are classified according to the number of its isoprene units, no? And so you have your monoterpenes, and so with the two isoprene units, and so your an example of this, your geraniol is this one is found in your essential oils extracted from plants, fruits, or flowers, no? And so that is what they call this. Each essential oil accordingly has a characteristic odor, no? And so, and they are used in making perfumes. Okay, and so again for your monoterpenes, and so they contain two isoprene units. Okay, and so example of that you have of monoterpene is a geraniol. No, and so of course monoterpene, and so do they have two isoprene units, and then of course with that they have 10 carbon atoms. No, and so please note that your isoprene units is a made up of 5 carbon atoms. Okay, and so here is an illustration of your isoprene unit and your isopentenyl pyrophosphate. Of course, and so this one, and so your terpenes, and so they are made up of your repeating isoprene units. Now, and so with this, and so again, as what we said earlier, your terpenes, they are not made, I mean, they are not synthesized from isoprene. Okay, and so instead, and so they are the synthesis of your isoprenoids. And so your isoprenoids begins with the formation of your isopentenyl pyrophosphate from your acetyl coenzyme A. Okay, and so this one is an illustration of your isopentenyl pyrophosphate. Okay, and this one is your isoprene unit. Okay, and so you have your examples actually of your terpenes, no? And so probably your isoprenes. No, and so you have here is your geraniol, and so this one is your monoterpene. Ayan. And the rest, okay? And so you have there, you have your limonene, geraniol, citral, your retinol, your beta-carotene, your menthol, your camphor, and the rest, okay? So this table shows you your terpenes, no? And so you have your monoterpenes, your sesquiterpene, diterpene, triterpene, tetraterpene, and your polyterpenes, no? And so with this, and so your example and the structure of each, okay? And so you have your number of terpene units present, and so they are your, you have here, okay? And so you have there. Okay, moving on, you have your keter, cis-keterpenes, and so your cis-keterpenes, and so you have here your terpenes accordingly that contain three isoprenes. Okay, and so your cis-keterpenes, and so these are terpenes that contain three isoprene. And so example, you have your farnesene, and so this one is a constituent of your citronella oil. And so this one is your farnesene. This one is used in soap and perfumes. No? You have there, and then you have your pythol, and so your pythol, and so it has four isoprene units. And so this one is, it has a four isoprene units, okay? And so this one is a diterpene, no? And so this one is a diterpene. And so note that yung monoterpene, and so that is, it's made up of two isoprene. And so when you say four isoprene units, and so present in a, what you call this, present in your terpene, and so you call it as diterpene. No, and so example of a diterpene, you have your phytol, and so this one is a plant alcohol, and again with four isoprene units. Your squalene, and so your squalene, and so is an example of triterpenes. And so with this, you have your six isoprene units. Okay, and so this is an intermediate in the synthesis of steroids, your squalene, and so intermediate in the synthesis of steroid, and found in large quantities in your shark oil, shark liver oil, and olive oil. And then you have your carotenoids, and so the carotenoids, and so you have your orange pigments in your plants. Okay, and so they are your carotenoids. Okay, and so they are actually your tetraterpenes, your carotenoids. Okay, and so tetraterpenes made up of eight isoprene units. And then you have your polyterpene accordingly. And so your polyterpene, and so they are high molecular weight molecules made up of thousands of isoprene units. And so example of your polyterpene, you have your natural rubber. Okay, and so you have there your natural rubber. Okay. Then you have your steroids. And so your steroids, they are derivatives of triterpenes. And so with four fused rings. Okay, and so derivative of triterpenes. And so if you say triterpenes, you have six isoprene units. Okay, and so they are found in all eukaryotes and a small number in your bacteria accordingly. Okay, ayan, so your steroids accordingly are distinguished from each other by the placement of carbon-to-carbon double bonds and various substituents present. And so like your hydroxyl group, your carbonyl group, and your alkyl groups. Okay, and so your cholesterol is an important, what you call this, molecule. And so an example of a steroid. And so this one is an essential component in your cell membrane, and this one is a precursor in the biosynthesis of steroid hormones, and then your vitamin D and your bile salts. And so they are formed from triterpene squalene. And so your cholesterol are formed from your triterpene squalene, and they are stored in cells as a fatty acid ester. Okay. Ayan, so again, ayan, so your cholesterol is formed from the linear, from your cholesterol is formed from your triterpene squalene, and this squalene is linear in form. Okay, and so you have there. And then you have your 7-dehydroxycholesterol. And so this one is the structure is similar to your cholesterol, but a double bond in the 7 and 8 carbon position is present. Okay, and so they are found underneath the skin together with your ergosterol. Okay, and so this 7-dehydroxycholesterol is your provitamin D3, or your a precursor of your vitamin D3. Okay, and so this is found underneath the skin, and this one is photochemically converted to vitamin D3. And so by the light rays from the sun, no, and so by the rays from the sun. Okay, and so your 7-dehydroxycholesterol, or your uh pro vitamin D3, or this one is converted to vitamin D3 by light rays from the sun. And then you have your ergosterol, and so your ergosterol, and so it is the same with 7-dehydroxycholesterol, but there is a slight difference in its side chain. And so this again, together with your 7-dehydroxycholesterol, this is found underneath the skin, and this one is converted to vitamin D2. And so again, with the aid of the light rays from the sun. Okay, and so converted to vitamin D2. And so you have here your ultraviolet rays, or the rays from the sun, and so will cause the rupture of the rings to produce this vitamin D2 and vitamin D3 from your ergosterol and your 7-dehydroxycholesterol respectively. Okay, and so just note a clinical significance with this is your vitamin D deficiency rickets. Okay, on your screens you have here your an illustration. I mean, an example of your animal steroids. No, and so you have your progesterone, your testosterone, and your estradiol. Okay, and so all of these are example of your human steroids. Okay, and so please note the structure of each. No, of your glycosides. And so your glycosides, they are steroid derivative still. Okay, and so they are what they call this your glycosides, or your cardiac glycosides. And so they increase the force of cardiac muscle contraction. Okay, and so your glycosides, they are carbohydrate containing acetals, no? And so you have here for your cardiac glycosides, and so it will increase the force of cardiac muscle contraction. Okay, and so accordingly, many of your cardiac glycosides are extremely toxic, and so but you have here few, and so are beneficial naman. Okay, and so you have several glycosides that are extremely toxic, and so you have your example of this toxic cardiac glycoside. You have your OABine, and so that is obtained from the seeds of your plant Strophanthus gratus, no? And so you have here, but then it has other cardiac glycosides. They have medicinal properties, and so examples of which, and so your digitalis, and so this one is an extract of a leaf extract from a digitalis plant, no, or your foxglove plant. No, and so a digitalis, and so this one is a stimulator of cardiac muscle contraction. And then your digitoxin, and so your this one, your digitoxin, and so this one is used to treat congestive heart failure, and so CHF, no? Ayan, so you have there. Okay. And so, and then your, what you call this, your your digitoxin accordingly is toxic. And so your digitoxin and your digitalis, accordingly, they are toxic beyond therapeutic doses, no? And so they are, what they call this, they are toxic beyond therapeutic doses, no? And so your digitoxin here will inhibit, accordingly, what they call this, inhibit sodium potassium ATPase. Okay, and so that is for your last. And so you have here your lipoproteins, and so you have here your lipoproteins. That is a combination of your lipids and your proteins. Okay, and so let's take a look at these lipoproteins. Okay, and so your lipoproteins, and so that your lipoprotein again, and so that is a protein that is linked to a lipid, and so you have your lipid groups, and so applied. And so this one actually is your lipoprotein accordingly is applied to molecular complexes found in the plasma of humans. Okay, and so your let's take a look at your plasma lipoproteins. And so, but then generally, your plasma lipoproteins, they transport lipid molecules from from one organ to another via through the circulation, of course, no. And then they also contain types of lipid soluble antioxidant molecules like your tocopherols and your carotenoids, no? And then, of course, and so your lipoproteins, okay? And so they are again, and so they are your apoproteins or your apolipoproteins here, and so that is the uh, protein component of lipoproteins. And this one, these apoproteins or your apolipoproteins, they are synthesized in the liver or intestine. Okay, and so with that, you have actually five classes of apolipoproteins. You have your apolipoprotein A, apolipoprotein B, apolipoprotein C, apolipoprotein D, and apolipoprotein E. Okay, and so with that, let's take a look at your lipoproteins one by one. And so this one actually is an illustration of a lipid. Okay, and so particularly your VLDL. No, and so this one is a VLDL. Okay, and so but then your lipoproteins, they vary in diameter, and so they range from 5 to 10 to 1000 nanometers. And so they vary in diameter from 5 to 1000 nanometers. And accordingly, each type of lipoprotein contain a neutral lipid core. And so you have that is made up of your cholesterol esters or your cholesterol esters or your triacylglycerols. And this core is surrounded by a layer of phospholipids, cholesterol, and protein. Okay, and then of course your what you call this the charge and polar residues on the surface of a lipoprotein, and so enable it to dissolve in the blood again, and so that is an illustration of a lipoprotein. Okay, and so your lipoproteins are classified according to their density. Okay, and so first on the list, and so you have your chylomicrons, and so they are actually the largest of your lipoproteins. And so it has a diameter of less than 1000 nanometers, no? And so it has a diameter of less than 1000 nanometers, but then it is the largest among them, but then it has the lowest in density. And so accordingly, it is a 0. 0.95. The density of this chylomicron is 0.95 grams per uh, cubic centimeter, no? Ayan, so you have there your chylomicrons, and so transport dietary lipid from the intestines to the muscle and your adipose tissues. Okay, and so remnants are taken up by the liver via endocytosis. Okay. Okay, and so you have there. And then you have here your very low density lipoprotein. Okay, and so your very low density lipoprotein, of course, in your lipolysis of your chylomicrons will produce your VLDL. And so this one is synthesized in the liver. And so this VLDL is synthesized in the liver, and it transports liver synthesized triacylglycerol to the tissues. And so it transports triglyceride endogenous, ano? And so it transports endogenous triglycerides to the tissues. Okay, and so you have there your VLDL. And so, of course, and so as your VLDL, and so as your VLDL are what you call this, depleted, or what you call this, nabawasan ng triacylglycerol and some apolipoproteins and phospholipids, and so they will shrink in size, giving you your IDL, your intermediate density lipoprotein, no? When VLDLs undergo lipolysis with that, and so it will form intermediate density lipoprotein, no? And so you have here your IDL again is produced from the removal of your some triacylglycerol, apolipoprotein, and phospholipids. And so please put a comma here. And this one is more dense than VLDL. And so it is more dense than VLDL. Okay. And then what do you call this? Further lipolysis of your IDL, your intermediate density lipoprotein, will produce your low density lipoprotein, no? And so this one is formed in from the lipolysis of your VLDL still. And so, but then this LDL, and so this LDL, okay, and so your low density lipoprotein transports dietary cholesterol to the tissues. And so it transports dietary cholesterol to your tissues. Okay, and so again, and so it transports dietary cholesterol to the tissues, no? And so that is for LDL. Okay, and so exogenous cholesterol. And then you have your high density lipoprotein, your HDL, and so this one is the smallest and most dense lipoprotein particle, no? And so this one is synthesized by both the liver and the intestine. Okay, and so you have here, it has actually the ability to what you call this, do your reverse cholesterol transport. And so your reverse cholesterol transport, or it transports cholesterol esters, and so from the peripheral tissues to the liver. And so again, this one being a reverse cholesterol transport, and so your high density lipoprotein transports cholesterol esters from the peripheral tissues to the liver. And so with that ability of your high density lipoprotein to really remove cholesterol from your peripheral tissues, from your cells, and so that is again called as your reverse cholesterol transport. And this reverse cholesterol transport is one of the main mechanism that explains the anti-iatrogenic property of your high density lipoprotein. Okay, and so you have there. Okay, and so again, and so it does your reverse cholesterol transport, and so other other description with that, and so you have your it scavenges excessive cholesterol from cell membranes. And so those items. Okay. And so moving on, let's take a look at uh this table. Ayan, so this one is a characteristic of a human lipoprotein. Okay, and so you have your, this one is your kilomicrons, your VLDL, very low density lipoprotein, your low density lipoprotein, and your high density lipoprotein, no? And so you have it at the density, sabi ko kanina for your kilomicrons at 95. And so, but then please make use of this, the table. Please make use of the table. Ayan, so at less than 0.93, no? And then you have your diameter, and so your lipid content, okay? And so those items. Okay, and so that is for your lipoproteins. Okay, and so you have here your relative mass of cholesterol. And so an illustration that illustrates your relative mass of cholesterol, your cholesterol esters, your phospholipid and proteins in your the four major classes of your plasma lipoproteins. No? And so with this, you have your lipoproteins. And so it is again your least dense, and that is it has a high content of your triglyceride, your low density lipoprotein, and then you have your VLDL and then your LDL. Okay, and so with this, it's your HDL, your high density lipoprotein, and so it has the highest protein content. And it has a low triacylglycerol content. Okay, and so please check on this. You have the blue one here, and so you have here as your triacylglycerol. Okay, and then you have the green one, you have your cholesterol esters, and and so you follow the color. Okay, and so that is for your relative mass of your lipoproteins. I mean the content of your lipoproteins.